My name is Michael Kravets. I'll be presenting digesting low-density polyethylene plastic bags using Pseudomonas Putida bacteria. Background. There are many environmental issues which this project targets, including waste management and recycling. In the past decade, there has been a large increase in global consumption of single-use plastic, including plastic bags and water bottles, and growth in usage of packaging materials. The lack of recycling infrastructure in many regions uh, has cannot sustain this level of waste. Also, the costliness of current recycling methods lead to the overconsumption of landfills, which affects local environments. In 2019, in the United States alone, 300 million tons of plastic went to landfills, and a large percentage of that flowed into natural habitats. The ultimate solution is to develop a cost effective method on a mass scale to process plastic waste without contributing to pollution and reducing stress on landfills. In this chart, we can see that only 9% of plastic is recycled worldwide, while a large majority of 70% goes into landfills, and 8% turns into pollution. Abstract. So in my project, I created 10 nutrient agar plates, and five were swabbed with the bacteria, while the other five were reserved as a control. So the plates were stored for 2 weeks in an incubator in a controlled environment of 24 degrees Celsius and 75% humidity and low light to simulate conditions in landfills, which are very similar to this. So after the 2 weeks, uh, the zero point 0.3 grams of shredded low density polyethylene plastic was applied to all 10 nutrient agar plates. They were masked before and then after the 2 months when they were in the same conditions. So the control lost an average mass of 0.022 grams, which was due to some moisture escaping, while the bacteria plates had an average loss of 0.092 grams. So the bacteria plates lost an average of 0.07 grams of plastic, which is about 23.3% of the total mass was lost. The results show that the bacteria was successful in digesting the low density polyethylene in these conditions, which can be simulated on a larger level in real landfills or recycling centers. So the materials used were colonies of the Pseudomonas Putida bacteria, was shipped from Carolina.com, the incubator at the 24 degrees Celsius, the shredded low density polyethylene plastic, which was sterilized under an ultraviolet light to prevent foreign bacteria from contaminating the experiment, the electronic balance to determine the mass, as well as proper lab techniques using sterile cotton swabs to transfer the bacteria, gloves and masks, as well as isopropanol to sterilize all the surfaces. Hypothesis. The null hypothesis was that the bacteria would not have an effect on the digestion of low density polyethylene plastics. And the alternative hypothesis, which actually happened, which was the bacteria will have a significant effect and digest a significant amount of the low density polyethylene plastics. Independent variables, the mass of zero point 0.3 grams of the plastic, the 8 weeks, which was constant for all the trials, the level of light, the identical colonies, and the identical nutrient agar solution, which was used for all 10 of them. The dependent variable which this experiment tested was the mass of the low density polyethylene plastic after the completion of the experiment. As we can see here, this in these two pictures. This is the plastic after the 8 week experiment. Results. So this is for the five nutrient agar plates with the plastic. We can see for each one, it lost a significant amount of mass, uh, about a 0.0 922. And for here, this is the control group. They also lost mass, but much less, and this was simply due to some moisture escaping. For this, some moisture escaped, and the plastic which was digested was converted into water particles as well as carbon dioxide, which escaped from the agar plates, which that's what caused the loss in mass. Analysis. So a t-test was created to show that the results were viable and the experiment had indeed worked. The variance is extremely small, which proves that the data is accurate, and the t-critical is slightly above 1.8, which shows that the plastic was digested and the experiment was successful. So, in conclusion, all 5 agar plates swabbed successfully digested an average of 0.07 grams. This experiment presents an ecologically sound way of treating plastic waste, and it could possibly be used on a large scale in the future. It provides an economically viable option as this bacteria is very cheap and reproduces very quickly. Uh, it's an option for waste management in the many third world countries that lack land for landfills or the resources to do that. Uh, so further study in this for this bacteria, it could be tested on a large industrial scale. In this project, we tested it. Uh, zero point 0.3 grams of plastic was shredded uh, per plate. This is a very small scale, but it needs to be tested on a large scale in a recycling facility or in a landfill with many tons of plastic. Also, the timing, we used a time of 2 months, but this should be used on a longer time of up to a year to see how much of the plastic can actually be digested in that period of time. And it should be quicker with more. It it should be exponentially quicker, so a lot more plastic should actually be digested. Also, there are many bacteria related to the Pseudomonas Putida bacteria. Find a similar bacteria in the same family. So the Pseudomonas Putida, it was never tested to digest plastic. So maybe another bacteria in the same family can have similar results. Uh, in the past, they have discovered several fungi and worms with these similar digestive properties, but I feel as though the this Pseudomonas putida bacteria, as it is cheap to grow and reproduce, and then grows in such common conditions with room temperature and high moisture, so it can deal with a lot of similar conditions, so it can be used in the future in landfills to solve one of the world's greatest problems of what to do with land and recycling these plastics, which are a major contributor in landfills. So this is my project. Thank you very much.